Okay. Hi, everyone. I'm Louise. I'm part of the OpenInsulin project.
And I'm Anthony, also a member of the OpenInsulin project.
And today we want to discuss one of our last blogs that we posted on the website about the medical supply chain and how with the OpenInsulin project we got involved in this, and we have uh, we were been thinking about different ways that we could change it so it's more efficient and more patient centered. Um, so feel free to ask questions at any time, and we'll be answer them. And in the meantime, I will just start with some questions just to go through the different point of the blog. So I think perhaps first, Anthony, if you want to kind of give us an overview about what are the characteristics of the medical supply chain right now.
Certainly. Well, we're mainly looking at the situation around insulin, of course, in the OpenInsulin project, but very quickly when you start to do that, you run into issues that are just ubiquitous in how medicine is made and distributed. And the problem for insulin is a very common problem, and it is that there are only a few suppliers in the entire world who are actually manufacturing and distributing many medicines, and there are far fewer generics as a result. There's far less competition in the market than you might expect from just about any consideration. Certainly the degree of need there is for these medicines. And it also means that the supply chains are very brittle. And when there's systemic disruption like the coronavirus epidemic, you see that because there are so few suppliers and it's so difficult to change how the supply chain works, it's very difficult for there to be an effective response to changes in demand. And when there's some sort of a disruption to transportation, for example, with the coronavirus situation we're seeing, there are far fewer flights of all kinds, including international flights, just getting harder to move things around the world and even just from city to city or state to state because of the need to limit people's travel to prevent the spread of the disease. Raises these really serious concerns about how people are going to get the medicine that they need to survive, like insulin, under conditions that are less than ideal, regarding all of the infrastructure that supports making and distributing those medicines. So in the OpenInsulin project, what we've been looking at, mainly in the case of insulin, but as we're going to discuss, it'll apply potentially to many other things. We've been looking at doing production at the smallest scale that is technically feasible. And in order to develop that idea, we've been looking at how medicines are made for clinical trials and how medicines are made in academic research, and just looking at the technology and the techniques that are used in those cases, and looking at how we could apply those kinds of small scale manufacturing techniques and technologies to insulin and to other drugs like insulin, biologic drugs potentially. So what we found is that it looks like it'll be pretty economical to set up production facilities that closely mimic how things are done in academic settings and for clinical trials, and then might just serve a few thousands or tens of thousands of people each. And we're looking at making the equipment that is involved in running those facilities open hardware so that we can avoid some of the unnecessary costs of proprietary equipment, and we're just looking at every aspect of how to make this economical. And as a response to the crisis in access to insulin, the main concern is cost. But coming back to these other issues that we're seeing with supply chains in the time of coronavirus, there will also be benefits to just resilience of the supply chain when you have, instead of 1 or 3 or 10 factories in the entire world for something, when you have thousands potentially. That'll mean that there will be much less need for transportation, and it'll be possible to make medicine much closer to the point where the people are using it are located. And there'll also be a lot more redundancy, so that if one factory is unable to operate for some reason, if it's regardless of whether it's a disease or a natural disaster, whatever the case may be, then one that is maybe just a few dozen or a few hundred miles away could compensate for that. So that's the general notion. And Louise and I, mainly Louise, worked on a brief blog post that discusses this in a little more detail. So we're looking to recap this a little bit and field any of your questions.
Yeah, and just to give to follow up on this, a lot of, even before the COVID crisis and pandemic, a lot of medicine are actually biological. So it's a case of the insulin, that is a small protein, but there's a lot of other illness that will be cured, or we use medicines that are proteins, and so that may be the case actually for a vaccine, like if antibodies are also protein. And even if you look at, so that's what we discussed in the blog. If you look at what are the main components of the testing kit, there are also protein, actually. The protein will take the RNA of the virus, will transform it back to DNA, and then amplify it so it's detectable. And so this is basically the idea behind all of the testing, is try to detect if you have RNA in your body. That means that you are infected by the virus. So, and we will need a lot of testing. We will need to test like half the population of the globe, probably, even, and different time, because we want to make sure, like, so we will need a lot of this. And for now, I think the protein are not on shortage. Some other part of the kit are actually on shortage. Not the protein, but they may get at some point. And also, if you have different factories that can do that, you're more independent about your testing kit, and you don't rely on just 1 or 2 company. And even when we will have a vaccine, we will need to produce a vaccine in very high quantity. So having small scale productions that can also be very flexible will help a lot. So when you produce protein, basically you're using the same equipment, whatever the protein you are trying to produce and purify. So you can imagine having some equipment that you can, if you have the protocol ready to go, in 1 or 2 months you can switch and perhaps produce another protein if you need to.
Yeah. So you mentioned a few of the components of a response to a viral outbreak like this that are proteins. Reverse transcriptase is one of them, and then when there's ultimately a vaccine that could have antibody components, or it could have protein components from the virus that might be modified a little bit to not be infectious or isolated from the other parts of the viral system. So we have a number of proteins that are involved in different phases of the response, from testing to treatment and prevention. So I thought it would be interesting to look at what difficulties have already arisen with testing. So for that, I believe it's the reverse transcriptase that is really important for part of those tests. And I heard that there's a very limited supply, that there's maybe only one manufacturer or just a few, and that they're overwhelmed right now. So is that something that you looked into more?
No, I didn't hear that, but I'm not surprised. So this kind of technique is widely used in public research and in research in general, but we don't use that like the same quantity that people are using now. So I'm not surprised that that will be the case, and especially when people get sick. Like if someone from this factory gets sick and they need to close down this factory, then there's no more production anymore. And so I think here's a problem. So for sure, having more factories, smaller, but more factory will help. And I think one also part that is very important with the OpenInsulin project is the open source part of it. And this actually also arises with a testing kit. Because so basically what during to run the kit, you could, like any academic laboratory will have everything, more or less everything to run it. But in general, you have what you call kits because like everything is very already prepared. You are very reproducible because they are always done the same, and it's easier to go through that if you are preparing all the solution by yourself. But the problem I think is like there's different commercial version selling by different company, and they were not always compatible. So if you develop an open source protocol, you can also think about this, like how I do my protocol so it's compatible, so people can modify, people know where it is inside, so they can adapt and change some part of it without having to just throw it away because they cannot use it anymore. And I think this is also very true for the hardware part. When you think about open source, is also, okay, let's think about the whole equipment, but also let's think about it as module that you can assemble. Like you don't need to have the whole thing to work, but you can just have perhaps one part and another part from something perhaps you already bought and from another company, but that's okay because everything will be everything compatible, easy to change and easy to repair. So you don't have to call the technician every time to check on this.
Yeah. I mean, that's really been a nightmare just in our lab. Is even if the equipment works, maybe the software version is just from a week later than it needs to be, and it's just a very slightly different version, and it will refuse to run, or you don't know how to configure it, and then it just gets worse for the actual parts of the hardware because all of it is just coming from one manufacturer, and they will probably not support something that is old and donated, and you then have to go looking for scraps, from other machines to try to repair your machine. And yeah, I mean, if we could have open standards, we've already seen firsthand just how much time it could save us and give us, so much more of a capacity to just rapidly adapt to what we're trying to do, instead of having every new piece of equipment take months or even years to finally figure out how to get it working.
Yeah, for sure.
Yeah, so I think when we do see the development of the open hardware take off, then we'll be drawing a lot of lessons from other open hardware projects and open source software projects, like in the biospace, OpenPCR has already been a big success in OpenTrons. So OpenPCR is a device that does DNA amplification by thermocycling. So it's a fairly simple device. It just has a temperature controller and a heater, more or less, and it it just takes the samples that you put in it between two temperatures, and this is something where the open source version just costs I don't know, a couple $100, maybe $400, and it runs on a standard microcontroller, and you can customize the software that runs on it all you want, and you can use a bigger heater or bigger cooling block or something if you want it to go faster. But the proprietary versions, a good one that performs well might cost $6,000 or $7,000. So it's not a small difference. There's like an order of magnitude savings in cost, and similar gain in the flexibility you have when you make something modular and have open standards for everything, and have the software and the hardware that controls it be open. And Opentrons is another case like that, where it's a lab robot that is basically similar system as a 3D printer would use to be able to position instead of an extruder, it's positioning a pipette to transfer fluids in a programmable way. And it's been able to ride this wave of technology that has produced really good 3D printers as commodities over the last couple of decades. And in that case, it's also taken machines that in the pre-existing proprietary economy could cost hundreds of thousands of dollars, and it's made them cost only hundreds or thousands of dollars. So that's actually something that we've been looking at to automate our high throughput screening that we're going to need to do to optimize the yield. So we've already started to look forward to being part of this open biohardware economy. But, as we continue to progress beyond the microbe engineering, we're going to, be setting up to make some direct contributions and add the protein production and purification systems to that. So the devices that we're looking to add are a bioreactor of a capacity in somewhere in the single digit to tens of liters. And that can actually draw pretty heavily on existing open source hardware for brewing. It's a very similar technical task that you need to accomplish between brewing and brewing organisms for beverages and brewing organisms for protein production. In both cases, you need to maintain a culture at a certain temperature and a certain level of oxygenation and various other parameters that affect the growth and the health of those microbes, and let them produce what you want them to produce. So there's already a fair amount of open source hardware for brewing, and we're starting to look at just taking that and adapting it to maintaining cultures of the microbes we're working with. And we're still focused on the microbe engineering, but to the extent we've been able to look at that, it seems it's just going to involve some minor modifications to that, where we might need to add maybe another sensor system that wasn't important for beverages, but will be important for us. But fundamentally, it's the same thing. And it's, again, somewhat similar to like the open PCR, where you just kind of, a lot of what you need to do is just maintain the right temperature and have something that can add and maybe, remove heat, and, a few other things that are similar. So it's fairly simple control problems, and at least at the scale that we're working at. That's an interesting side note, I think, is that the smaller a batch you have, the easier these process control issues become. So there's a pretty serious diseconomy of scale in having a 4,000 liter bioreactor or something like they would use in a big factory, because then just the problem of keeping the whole thing at a uniform temperature is a really challenging technical problem, because how do you, if you have your heaters at the bottom and it's 30 feet tall or something, how do you make sure that that's all, even in temperature? So then you need a really effective mixing system, and you might need more sensors, and, just adds a lot of complexity and sophistication, where if you're working at a smaller scale, you don't have to worry as much about these kinds of nuances. So we're looking to, build on this and pick up some of these gains from just working at a more appropriate scale and scaling out instead of scaling up. It's kind of a similar lesson to what happened in, computers when they moved from mainframes to clusters of commodity personal computer hardware. Turns out that, there's a similar effect in the engineering, where the complexity of making one giant computer is a lot more relative to the performance you get than just having a bunch of smaller computers and networking them together and letting them work together.
If we when you look at like pharmaceutical, how they develop drugs from, identify the target to commercialization, actually, like the scale we are looking at, that is like 10 liters, they still consider that like research and development is still at the lab. For them, it's just testing, because they will go up to 20,000 liters sometime for some production. So what we are looking at is very like at the beginning, so it's very close to what you will do at the lab. So this is so that's also great, because we have a lot of people working with us who have a background in public research, and it's very close to what you will do in an academic setup. Is a little the same. Just we will need to adapt our protocol to fit all the regulation, because it will be a medicine. But even this, because it will be so small, so you can imagine like everything that Anthony talked about. So the bioreactor, then the FPLC, so to purify the protein, and then the formulation could stand in one like dining table. So it will be quite small. So you can imagine even to have like a tent to sterilize all, like to have a sterilized environment. So anything you want to do around will be easier, just because it's so small. And something we're also considering is try as much as possible to automatize the whole setup, so it's a less human interaction. So it's really like just one process. We see if we get to that. But it will be very, very small, and also because it will be small, we also be very flexible. So you can imagine producing insulin, perhaps producing different form of insulin, but also any protein you may need for like if it's in a hospital setup, any proteins they may need for the patient.
Yeah, and coming back to that, so you mentioned again like reverse transcriptase for testing, and beyond that, I was trying to find a little bit about vaccine work and treatments that people are doing. So I've obviously that's not done yet. No one knows what is going to be the treatment or the cure or the vaccine for this. But I found that there are a few classes that this falls into. One would be like taking a viral protein, like something from the code of the virus or something, and just making sure it isn't functional, and then producing a lot of that and using that as a vaccine so that your immune system will react to that protein code. And then another thing was like making the antibody itself and using that as a component for a vaccine.
Also, so this will not be a vaccine, but what happened with patients when they get infected with a virus is some of the like second effect will be an immunitarian response. So basically, your immune system reacts too strongly and start like it's just over the. So you have high fever and So you will have also medicine. I think they are used for other kind of virus. I think they name interferons. They're also protein, and they just try to calm down your immune system so you don't overreact. So this is not really like taking care of the virus, but is basically taking care of the second effect of the virus that can also be deadly. So you probably won't have. So I mean, you will have the vaccine, but before that, you will probably have a large range of different medicines that are for different, like depend of what are your symptoms and what is what perhaps step of the infection you are, and probably some of them will be proteins. And in this case, you will need a large amount. And also, what we already see happening is so first is accessibility. So you need to produce enough for everyone, and also who produce who. So this is also like who owns the patent, who produce, and how we decide who get it. Who get it first. So if you have a lot of site of production and you can produce first, you can produce faster, but also the different state or hospital or organization can also produce their own, so they don't depend in other organization or other company, because that will probably happen with the COVID, that the people producing the medicine or the vaccine may sell it for people who can pay.
Yeah, definitely an issue we're seeing with insulin right now.
Yeah, you're already saying with insulin. I think insulin was a great of what can happen. So yeah, you want to be able to always be sure to always be able to provide for your patient. So you want to be able to produce by yourself. And what we heard a lot, especially for biologics medicine, is that this is complex. And that's true, it's complex, but it's not that complex. Like you need to work on it, you need to follow regulation, and you need to learn how to do it. But we can imagine doing it in hospital setup or small setup, as is done in academia, just with more regulation because it's a medicine. But the technology that they use to produce a biological medicine has been around for like 40 years. It's not rocket science. A lot, like I have a PhD in biochemistry. I know how to produce protein, and a lot of people who have a PhD, even a bachelor degree in biochemistry, could do the same.
Yeah,
So
well, that's something I noticed when I was looking into this and where this is going on already. Whenever a biologic drug is made for a clinical trial, it's made at a fairly small scale, just for a few hundred or a few thousand patients, or sometimes even less. And clearly that has to be safe, and everyone wants that to be absolutely safe. Probably for a profit motivated corporation, they're going to have even stronger incentive to make the drug safe for the clinical trial, because they don't want the trial to be influenced by safety issues in the drug itself. So clearly the means to make medicine that's safe to use at that scale already exists and is already in common use. So it's not really a question of making some sort of a breakthrough to make that possible.
And a lot of people also are concerned about safety. And some thought I have about that is, actually, if you go for small scale, so what we call a batch, so you will have, like, you fill up your bioreactor and you let it grow, and for any reason, when you go to and then you get it out and you put it on your purification system, and for any reason something go wrong, and then you have so small quantity, you just you realize it's wrong, something it got contaminated or something, you just throw it out. It's a 1 week work, but and it's so small volume that you don't care, you just but if you have a bioreactor that in 20,000, you have been growing that for like perhaps 1 month, the incentive to just throw it out actually much less. So so actually, if you go for small scale, you can be much safer because you can be much more strict about your safety condition and what you consider to be pure and what you consider to be correct. And so I like the idea to have
Yeah.
small scale production, like, people can visit, can see how it's done. Perhaps, no all the time when it's shut down sometimes, but and perhaps can access like all the transcript, like all the record, what like the number of the batches and what happened to them, and so people really like patient know what they inject in themselves, and can really trust the whole process and even learn it if they want to know, how what I inject in myself every day come from.
Yeah, I mean, I'm getting inquiries on our project email just about every week from people who, have diabetes and they're just curious how insulin is made. And, often just today, someone, maybe yesterday, sent an email and said, right now I'm just kind of curious how it's made, and maybe in the future I'll, try to set up something local where I'm at to make it. But, I just want to know right now so that I can tell my friends just how this all works and understand it for myself. And that's something that you definitely don't get with the current system. You can't just go to the factory. You can't talk to someone who works at the factory, really. You just have to accept this, magical little container, and can, if you are really sophisticated, then maybe you can go and look up the papers in the journals if you can get journal access. And then even then you're just getting this kind of high level view of how it's made. It's not going into the details of, how the equipment works and the basic techniques that are involved that the author of the paper is just going to take for granted everyone knows. So, this is really going to just at a very basic level make it much more transparent to people, like, what our lives depend on, how it works, what it really takes to make it, what all the trade-offs involved in that process are. Then you can, then we can start talking about it with many more people and, seeing how to do it better.
Yeah. I was also reading about the formulation process and how, depending on what when you have your protein that is pure, sometimes you want to add some additive to make it more stable, or you can actually slightly change the activity of your protein if you depend on what you add, and this formulation process, so I think it's actually patented too. So you don't always know when you use like commercial insulin what is the formulation, and perhaps sometimes when you change from one insulin to another, if the formulation is not the same, I think it may trigger some immunity response too, depending of what they had in the formulation. So also having transparency about, okay, this is the way we do the formulation, this is all we put like inside, and this is like the same that another perhaps insulin, commercial insulin, or even having different kind of formulation depending of what the people need, but with the idea that the small scale production serves the patient, and whenever the patient says that, for example, this make our life easier, let's do that, you can completely adapt your product to the patient, and not the other way around. The patient have to adapt to a product anytime that the farmers decide to change something on their product.
Yeah, there have been a couple cases that were really well publicized a couple of years ago, and I'm sure there are many more besides the ones that were just publicized. But there was someone, it's like an 11-year-old girl in Arkansas or something, I think, and she was allergic to one brand of insulin, but another brand worked fine because of some small difference in the formulation, I think. So This was only a problem though because her insurance was through her mother's job, and that insurance would only cover the one she was allergic to at the best price, because they have all of these, often negotiated and secret arrangements to just have exclusive, distribution arrangements, so that your insurance will only cover one of the three brands at the top tier of coverage where it's actually affordable. And then if you want to use another brand, then you have to pay, tier 4 coverage. You have to pay like 80% of the list price or something, or worse. And the list price is so inflated that this means it goes from maybe $100 a month to thousands of dollars a month. And it's just insane. So now it's like, okay, pay $20,000-$30,000 a year just to stay alive because you're allergic to the one that your insurance company covered. And it's just completely arbitrary. It's nonsense. And this, as you mentioned, this could be something where, if you identify that component in the formulation, maybe you just omit that and you say, okay, here's the insulin you need. Sorry, you're allergic to this additive or whatever, and maybe you have to keep it in the refrigerator more, but you still be able to have the insulin you need for a reasonable price, instead of suddenly having to buy yourself a new car every year to stay alive, the equivalent price. It's just and then what, so many people can't do that, and then they end up dying, so for absolutely no reason.
Yeah, this system will be more accessible and more flexible for patient, and also easier to adapt. Like if so there've been new insulin, like they do a mutation, they try new stuff, they add the lipids on it, so to get like faster acting insulin or slower acting insulin. So of course, I mean, this is a patent issue, but if the old protocol was open source, you can also produce this new insulin in a few months. Like you can just go on it, and so the patient can get it quickly once it passes all the FDA approval. So it's also a way to get a medicine to patient. I also want to mention, so now we are talking about insulin that is like several lot of people. But this small scale production could also be used to produce protein for disease that's just concern like a few handful of patients, perhaps 50 over a country, but producing in a way that actually those cost too much. And it's actually a problem because if you have an illness that knows that many people have, there will be very little research about it, and even little production of medicine, because it's not profit. The pharmaceutical will not make profit out of it. So I think there's also this all wide range of, like, we know what can help the patient, perhaps not cure them, but at least help them in their day-to-day life. And we may, this equipment can be used to produce this protein in a very fair price, like in a hospital. And this happened in the Netherlands for 1 or 2 years. They actually produce themselves, they come up with a protocol and then produce themselves a protein and what injected to patient in a kind of clinical trial test, because they just have like 50 people with this illness in Netherlands. So you can also imagine that. And then if you want to go to the next step, is also if you can produce, if the hospital has the possibility to produce the biological medicine, they don't want to produce all of them. There's a lot. But if they have the equipment to do so, then when you go and negotiate prices, so not that much in the US, but ever outside the US, the country in general negotiates the price of the medicine, then you have much more leverage, because you can always, leave the table and say, you know what, this is way too expensive. I would just produce myself. I would just put my biochemist on it, and in 1 year I have a protocol running, and I will just produce it. So this also may help to reduce the expense of healthcare system, who will be able to negotiate much better prices. That's what happened now.
Yeah. Yeah, I mean, there are a lot of diseases where it might just be a genetic disease where you can't make a certain enzyme, and we know what the enzyme is. Someone has, in an academic setting, figured out how to make it in a form that will be an effective treatment. But just because there's only a few hundred or a few thousand people with this who are lacking a functional gene for this thing, then they're just say sorry, but they made it in the academic lab to do the research, right? So you could run a similar facility, and there's no reason you need to set up $1 billion plant to do that, right?
Yep. Especially for small population, you can imagine to treat them with like the same setup like clinical trial, and sometimes you can actually cure like your population, because it's so few people. With clinical trial, you don't even need to. I mean, then we are done. Like the medicine can be in the market if you want to, but you can actually serve this patient like right now with this kind of protocol. And so perhaps you heard that. I just wanted to mention it. One of the very hot topic now is precision medicine, and perhaps you are wondering if that is related. So it's slightly different. Is the idea that now so we learn more and more about, I think especially with cancer. So with cancer, we realize that it's just not illness. It's a lot of different ones with a lot of particularity, and is related to your genetics. So depend on your genetics, depend on a lot of different factors. And now more and more we are able to kind of put the different kind of cancer with different groups, and there's different way to deal with different kind of cancer. So you can have the X-ray that is kind of killing all the cell, but then you can have very more subtle way to attack the cancer cells, and it will depend of where your group of cancer. So having a way to cure very specific disease, like a very specific cancer, this is called precision medicine. And in this case, even the pharmaceutical company realized that now you cannot go with this big plan, because it's too much, and you just want to produce a very few amount of them. So there's also this direction to go, so they call it precision medicine, that you will just produce a few amount of your medicine once you identify what I think is very true for cancer.
Mm-hmm.
Like you just identify this. So also like small setup. I think it's still bigger than what we are talking for us, but it's not like large brand production.
Yeah. Well, one of those is what, like CAR T immunotherapy, where you actually need to produce an antibody that is really specifically tailored to one person's specific body and their cancer, right?
Yeah. And then you also have, then this is all like the stem cell therapy, but this is very, very precise, like if it's just for one people. But I think it's the same also thing. The same is they will have to do like small batches too. So probably there will be a lot of technology. It's just like what we want to bring is open source technology that everyone can use it. And always think, okay, how I can do this the easiest way possible, that I will not just use this technique just because, I like it. You have to make sense, you have to be easy to use and cheap. So for example, one example I can give is a way to detect protein is to use UV. And in general, what was used was a UV lamp, but that's quite expensive, would be at least I think $1,000 just for your UV lamp. And so we are exploring to use LED now that do like UV LED. So we are exploring to see if the detection level is good enough so we can detect our protein with that. But always try to get to find other way to do stuff so we can come up with an equipment that will be at the end for all of it like $10,000. And the same, like easy to repair, easy to do, and really with just the equipment we need. Okay. Do you want to add perhaps a final thought, Anthony? Then we can wrap up.
Oh yeah, well, like everyone, we're just kind of trying to keep our work and our lives going in less than ideal situation, and I just want to wish everyone well and offer some solidarity, and also invite people who might be interested in joining us and working on some of these things that we've mentioned to join us at one of our meetings online. And also on Sunday, the 26th, the Sunday following tomorrow, we're going to be having a new member orientation meeting at 10:30 Pacific on our usual video conferencing channel, same one we're using right now. I believe meet.jit.si/OpenInsulin. And so we will be publicizing that more closer to the time of the event. And I don't know, Louise, maybe you could mention what the best ways to keep in touch with us otherwise are also.
Yeah, sure. So it's great to just step during our meetings. They are all open, so you can just come and see. You can connect with us via social media, and I can also put our email. I will put it on the description with also the date and time for the new member meeting. We don't have access to the lab right now, so our science team working on the web lab in general, like in usual time, are meeting at 1 p.m. Pacific time. And now what they are doing is that they are discussing a publication and paper about like how we produce insulin or how we provide what we do, like this technique and not this one, what we use this strategy or this other one. So if you are curious, if you want to know more about how we produce insulin, you can also join us. So it's every Sunday at 1 p.m. and I will also add it on the description of this video. Yeah, thank you all for watching that and attending. And if you have other questions, I know sometimes questions come up later on, you can add it in the comment and we will answer them. Okay. Yeah. Thank you for attending and stay safe.
Yeah. Thank you.